into the lecture series on cell culture technology. So, today we will be doing the 5th lecture of this 5th week. So, if you guys recollect, in the last lecture we talked about, so we took a case study that a company has to screen few drugs which are believed to help Alzheimer's patients, and hypothetically in a virtual world we set the quantification parameters as, by the application of the drug, the number of synapses will increase, or in other words, synaptic button could be quantified in that way. And while going through it, we kind of realized that there are few aspects which demands development of a much more advanced model system, that too at the in vitro level. So, the point, just if you recollect, we came to the point that we needed one point which came out very clearly in front of us is we needed a pattern network, a network which follows a information transfer in a direction. Second, we may opt for developing network in 3 dimension, from 2 dimension to 3 dimension. Okay. And third emerging concept which kind of made things to think or revisit is it a wise idea to use a fetal hippocampal culture, which is fetal day 18 out of the 22 to 23 days of pregnant mother from where we derive the fetus, the 18 fetus. Is it a good model system to evaluate Alzheimer's system? So, that brings us to some of the futuristic search for solutions. So, the futuristic search for solution starts with the core goal is we wish to have a 3-dimensional pattern network of adult hippocampal neuron as a test bed. So, let me just put it together. So, we are into week 5, lecture 5. So, desired goal is 3-dimensional 3D patterned network of adult hippocampal neurons, 3D pattern network of adult hippocampal neuron as a test bed for screening drugs, or you can say it potential drug candidates against Alzheimer's disease. And of course, in this case we will be using the possible model system, animal system could be three possibilities. To start off with a rat, mouse, and if possible, if situation permits, then going for human neurons from obtained from surgery table. Okay. So, your first cut will be A or B, and of course, the third one will be C. And we will talk about it, how you can obtain the human neuron. Especially this can happen if, say for example, there is a brain tumor surgery which is going on. So, when they remove the tumor from human brain, one of the thing which happens is that a part of the good brain tissue kind of is being also lost. So, if you have the skill set and if you have the technical know-how to collect that tissue from the operation theater and process it, and of course, use that particular part of the live neurons which are present there, you actually can have a human neuron culture on a dish. So, we will come later into that. So, let us think for, or let us explore how from. So, the first thing is you have to, now your source is going to change. So, your source is changing all the way from fetal to an adult. So, if you remember, when we talked about a fetal hippocampus, it is more like this. It is a kind of a very soft F18 tissue, whereas in the adult tissue it will be much more bigger. It will have something like this. Now, this tissue as compared to the fetal one cannot be dissociated by mechanical route. So, in the case of fetal, you can mechanically dissociate the tissue. Okay. On the other hand, if you want to develop an adult cell culture of adult hippocampal neuron culture, you cannot do so by mechanically. You will be needing, first step will be, prior to this will be enzymatic, followed by of course mechanical. Now, when we talk about enzymatic step, what enzyme will suit, because it is an adult tissue. So, the enzyme has to be, I would say, has to be mild enough not to damage these cells, because the regenerating potential, these adult hippocampal neuron regenerating potential is very, very limited. Limited ability to regenerate. Okay. So, if you know it has a very limited ability to regenerate, limited ability to regenerate. So, if you know it has a limited ability to regenerate, you do not want to damage this tissue extensively. Okay. So, for enzymatic treatment, it has been observed the enzyme which is reasonably better than other enzyme is called papain. At a fairly low dose, and I will give you an additional reference where you can look that how papain, or rather what dose of papain will help you. So, papain acts as an enzymatic dissociator. Okay. Now, what you do in this case, you have to optimize the time. Not only you have to optimize, so your first optimization comes here, quantity. Quantity optimization. Next thing which comes is time. Second thing in the same line, temperature. Sometime you may need to raise the temperature, but you have to be careful whether you can do it in regular, or you want to do it in a water bath of say like 4 degree, or you want to do it at 20 degree. That decision you have to figure out over a period of time of optimization. So, once you optimize these parameters, then you have to realize while you are mechanically dissociating a fetal tissue out here in this situation, in that situation most of the prominent cells which were present there were pyramidal neurons and emerging glial cells, which are mostly astrocytes. Now, one of the thing which over the years cell culturists have done, especially those people who are good at neural cell culture, they have optimized the media for neurons, optimized medium for neuron. What does that mean? Now, that means that the medium which has been developed selects or preferentially allows the neurons to grow better as compared to the glial cells. So, that essentially brings us to a point. Of course, we know in the brain you have both the glial cells and the neurons, and glial cell outnumbers the neurons, and glial cells have multiple functions. If it is an oligodendrocyte, then it supports the myelination. If it is a Schwann cell, it supports the myelination outside the central nervous system. If it is an astrocyte which is present there, they will support the homeostasis. It will ensure that excess neurotransmitters and neurotransmitter related toxicity are being avoided. So, when we talk about developing this medium, one has to make a call that do you want a mixed culture, really absolutely close into real life culture. But then if you have to do this, there is one catch. Catch is this, that neurons may not divide, but the glial cells will divide at a faster rate, and if you allow the culture for a prolonged period of time, then the glial cells will outnumber the neurons. In a big way, outnumber in the sense will be competing, because they will be dividing, they will be consuming lot more nutrients, and the culture will eventually become overpopulated, and you won't be able to keep the culture for a prolonged period of time. So, you have to think of an optimized way if you want to do a mixed culture, that how you can ensure that the population of glial cells are kept at check, that they do not proliferate so much so that you cannot really handle the culture. But having said this, let me tell you, if you could really mimic the exact condition where you have small amount of glial cells attached to it, then you are much more closer to the real life situation, and it also influences the electrical activity of these hippocampal neurons. So, the mediums which are commonly used for fetal hippocampal neuron are called neurobasal medium. You can go online and check it out. Okay. Neurobasal medium, and neurobasal medium is supplemented with a composition called B27. This composition was developed primarily by a gentleman called Gregory Brewer. Okay. At that time, he was in Southern Illinois University at Springfield, Illinois. One second. Yeah, Gregory Brewer, Southern Illinois University. Initially, he developed something called B10 or 6, something like a medium, and he further took it, make it B27. So, B27 stands for 27 different components which are added to it, which primarily supports central nervous system and other neuron growth. Of course, later it has been proved that it also supports the growth of other excitable cells like muscle cells as well as cardiac cells, and that was much later it was proved, but initially it was developed to support the growth of especially the hippocampal neuron and other brain neurons. And you have to supplement. So, neurobasal is being buffered using sodium bicarbonate, okay, sodium bicarb buffering. So, you have to use the CO2 incubator, and most of the CO2 incubator prefer 5% CO2 for embryonic hippocampal or any of the hippocampal neuronal culture. This is supplemented by antibiotic and antimycotic, and there is another component which is added, which is called glutamax. Glutamax is nothing, it is basically a dipeptide of glutamine. You needed to add glutamine because this is one component which goes bad very fast. So, what at one point Life Science Technologies, what you know of, initially it was in vitrogen, they developed a product where 2 glutamine, it's an amino acids, you couple 2 glutamine using a peptide bond, you make a dipeptide, and that is called glutamax. Apparently, it has been observed that glutamax is a much more potent stable compound as compared to using glutamine, which otherwise is not really very supportive. I mean, it supports, it's just its shelf life is very less once you mix it in the medium. So, this is the medium which was developed mostly in this field. You will see most of the work, very initial pioneering work were done by Professor Gregory Brewer. So, and his whole idea was to have, I mean till this date the kind of work he publishes, you will see more of a in vitro analogues for testing different kind of Alzheimer drugs and all those kind of things. Okay. So, this is the medium which is used for embryonic or fetal hippocampal culture. Now, the modification what has been made in this medium is pretty much all the components remain the same, except its osmolarity has been varied, because the osmolarity of this medium is approximately 215 to 225 milliosmol. Okay. Whereas, the osmolarity which you needed for adult hippocampal culture is osmolarity of the medium is around 260, so 270 milliosmoles. So, this value has come from cerebrospinal fluid of an adult rat. Okay. So, the next advancement in this medium which was used for fetus is using neurobasal, but it comes in the market, it comes with A, where A adult stand for adult. So, neurobasal A and neurobasal regular, the difference is in osmolarity. If I exactly recollect, there is a higher concentration of sodium chloride in neurobasal A. So, essentially you can have a neurobasal regular and you add that prerequisite amount of NaCl to make it neurobasal A. Okay. So, this is how from neurobasal you convert it into neurobasal A. So, this is, as a matter of fact, in the course, this is the first time I am introducing a medium, a specific medium. Okay. So, medium remains more or less the same, except that now you have to introduce another step which is called enzymatic dissociation, which is the papain. Okay. Now, this dissociation, which again the work has to be referred to Gregory Brewer, he optimized the condition. He found that this dissociation more or less takes around 30 minutes. Now, at 30 minutes, we dissociated all the cells of hippocampus, and this composition consists of your adult hippocampal dissociated cells. What all it consists of? 1. Neuron. Within neuron, pyramidal neuron 1, interneuron 2, okay, 2, glial cell, astroglia, making astrocytes, oligodendrocytes, which most of them dies out, but you will still have contaminants which are function as myelinating cells. So, what you are getting here, think of it, you are getting a mixture of different cells. So, your next challenge, how to separate cell separation. So, you see now slowly we are moving with one example, we are moving to the complexity of cell culture. So, I will close in here. So, next class next week we will be starting to some of the basic things, again following up on this case study, how one can separate different cells, and this paradigm will remain the same, only the cell type will vary. Thank you.